everyone, and welcome to another North Carolina Botanical Garden Lunchbox talk. Today, we're excited to bring you Jess Roach and Dr. Darren Penny from UNC Wilmington. I'm Joanna Massey-Lalikas. I'm the Director of Learning and Engagement at the North Carolina Botanical Garden here in Chapel Hill, North Carolina. And we're really thrilled to offer today's program in partnership with the Friends of Plant Conservation. I'm joined today in this Zoom room with David Michaud. He's our Registrar and Program Coordinator, and he's here to help answer any Zoom technical issues that arise, and will assist our speakers with Q&A at the end of the talk. And we have folks tuning in from across North Carolina today, so thank you all for joining us. So we're excited to have Jess with us today. And Jess Roach, she's pursuing her master's in biology at UNC Wilmington under Dr. Darren Pennies, who I believe also is joining us today. Conservation biology became a passion for her as an undergrad at UNCW while propagating Venus flytraps and tissue culture. Through that research, she became familiar with the unique nature of the southeastern coastal plains and its rich biodiversity, leading to her current pursuit as a graduate student. Jess is pursuing a career in conservation biology. And I'll also mention Dr. Darren Pennies is an associate professor of biology at UNC Wilmington. His research focuses on systematics, natural history, and evolution of angiosperms. And he's a global leading expert of melastomes. Excuse me. So thank you for joining us today, Jess. I'll hand it off to you.
Thank you, Joanna. Thank you so much for the introduction, and thank you for hosting this today. Looking at the participants, I'm already sweating. I'm a little bit nervous. This is the big group that I've been waiting to say things to for some time, including my advisor, Darren Pennies. Thank you so much for being able to be here today. And one of my committee members, Johnny Randall. So many of you know who that is. Thank you for being here as well, Johnny, and I see Mike and a bunch of other big names in there. Natural Heritage Program, Alan Weekly, thank you all so much for being interested. I will try to give you some stuff that is of interest today. Okay, so let's get started here. Of course, as Joanna has already told everyone, my name is Jess Roach, and I am a grad student at UNC Wilmington. I'm conducting a population genetics study of Lysimachia asperulifolia, who many of you are very familiar with. And studies like this aim to identify genetic variation within populations of individuals through space and time. But more on that shortly, right? So first things first, right? Let's talk about where we can find this plant, right? When we think of places with high rates of biodiversity, we tend to think about tropical and extreme parts of the world, such as New Caledonia, Caribbean islands, the Mediterranean basin, places like that, right? However, it may surprise you to know that the southeastern coastal plains region of North America is recognized as a global biodiversity hotspot. This classification is defined as having more than 1,500 described endemic species confined to a region with greater than 70% habitat loss. So something you'll notice, I'm over here in Wilmington. We have a nice peak in biodiversity right down here. And as many of you know, again, we are nearly subtropical, and we have some really interesting geology that has led to this rich biodiversity. So looking at that region, you'll notice that the coastal plain floristic province lies within that geological southeastern coastal plain, and it's host to several ecosystems that are unique to this region, including the longleaf pine savanna. And longleaf pine savannas once dominated the southeastern coastal plains, covering about 90 million acres, which is roughly the area of California, from Virginia all the way through Texas and the southeastern United States. Perhaps not so surprisingly, humans have caused this vast ecosystem to decrease to less than 5% of its historic range as of today, amounting to approximately 2 to 3 million acres. Longleaf pines produce an assortment of valuable resources that colonial North America egregiously exploited, beginning in the early 1700s. And nearing the end of the 1700s, North Carolina in particular became one of the world's largest producers and exporters of naval stores, including tar, pitch, and turpentine. These are all products that are used to build and maintain ships, as we know. There were many wars around that time, so we needed ships. They were one of our biggest assets in winning wars, right? So other anthropogenic impacts and disturbances that have led to the decline of this of the longleaf pine savanna include lumber harvest, fire suppression, which is still ongoing today, but we are trying to mitigate that, hydrologic change, agriculture, and urbanization of the land. Higher than expected rates of botanical endemism and rarity occur throughout this region. And if you're looking at North Carolina in particular, it can host more than 20 endemic plant taxa in certain regions, defined today mostly by county boundaries, as you can see here. And where we are down in Wilmington, this is New Hanover County, this is where we are. You can see we have areas that can host 20 or more different taxa. You guys too, you're not too far off, right? But pay attention to this red, right? These are the very high areas of rarity and endemism. All right, so continuing on, right? As the name would suggest, longleaf pine savannas stands are dominated by longleaf pine, while the herbaceous layer is characterized by the presence of wiregrass and associated graminoids. There's abundant diversity in this herbaceous layer, and 1 square meter can host as many as 50 different species of plants, which is crazy if you think about it. Within that highly diverse layer exists some of the most charismatic and recognizable plants, many of which evolved here and are endemic to the southeastern coastal plain and North Carolina. This encompasses 5 genera of carnivorous plants, including the very sexy monotypic Venus flytrap. We also have more than 20 genera of orchids, and the plant of focus in my research, which is Lysimachia asperulifolia, or commonly called Pocosin Loosestrife. So let's go back to the things that we know, right? Perhaps the most recognizable plant, we've narrowed this down to ecotones at this point, right? Would be the Venus flytrap. It's often found along ecotones of longleaf pine savannas and pocosins, which are just upland wetlands. And to explain an ecotone, those are just transitional zones of vegetation between 2 different plant communities, which can contain some of the characteristics found on each bordering community and often contain species that cannot be found in the overlapping communities, such as Venus flytrap or Pocosin loosestrife. Flytraps in particular are narrowly endemic, and they're only found in the coastal plain of North and South Carolina. They thrive under strict habitat requirements, including mesic, nutrient-poor acidic soils with frequent fire and an open understory. The current consensus indicates an approximate range of about 75 to 100 mile radius around Wilmington in particular. And it goes across 18 counties in North Carolina and 4 in South Carolina. So it's a pretty small, that's why it's a narrow endemic, right? Pretty small range. We don't currently have federal protections for this species, but a formal application submitted in 2016 is under review, as most of you know. But what about all of the other rare endemic plant species I just mentioned can happen in this ecosystem in the longleaf pine savanna? We are fascinated with Venus flytraps due to its animal-like behavior, and it has these atypical life habits that tend to overshadow the importance of many other, perhaps less charismatic, but even more imperiled plants. So what I'm referring to is my study organism, right? Lysimachia asperulifolia. This loosestrife is a narrow endemic, just like Venus flytraps. It's commonly found along the ecotones of longleaf pine savannas and pocosins. If it's a narrow endemic, it's defined as having a highly specific form of rarity, where those species only occur in one or a few small populations and are geographically restricted to those localities. It shares nearly identical habitat requirements as the Venus flytrap, and observation of one species is often indicative of the other. So Lysimachia asperulifolia has known populations in 11 counties across North Carolina and 1 county in South Carolina. This loosestrife is listed as endangered under the Endangered Species Act. It's vulnerable in North Carolina and globally, and imperiled in South Carolina. Federal protections and active monitoring programs were proposed in April of 1986, then approved in June 1987, and have been enacted since July 1987. NatureServe currently ranks Pocosin Loosestrife as vulnerable in North Carolina, critically imperiled in South Carolina, vulnerable at the national level, and vulnerable at the global scale, with a need for a new status review since the last review was in 1993. All right, so more about this plant, right? Pocosin loosestrife is self-incompatible. We've known this for a while. It's a clonal organism spreading through underground stems called rhizomes. Rhizomes in these plants are presumed adaptation to the frequent fires that occur in this region, which in combination with low nutrient availability maintain the low shrub layer that they need and prevent a hardwood understory from developing. Many rhizomatous species tend to produce vegetation rapidly after burning, and fire is known to promote flowering and fly traps and other fire adapted plant species like wiregrass. This is not the case for loosestrife. In fact, loosestrife does not typically produce flowers, and when it does, fertilization events are rare due to lack of effective pollinators and suspected sterility and/or S allele incompatibility. S allele incompatibility occurs when the genotype of pollen matches with one of the S alleles of the receptive female aspects of the same flower. And self-incompatibility is an effective mechanism, right, that's promoting outcrossing in plants and is the botanical equivalent of avoiding incest. Thus, those self-incompatible specimens with the same S alleles are inter-incompatible and are allocated in the same incompatibility groups. But more simply, loosestrife is a clonally dependent organism with an inexplicably poor sexual reproductive strategy and has not exhibited population increase nor stability in monitored populations over the last 30 years. In fact, results from 10-year US Fish and Wildlife Service monitoring and management plans have shown an overall decrease or undetermined status of these populations with by using a population viability analysis using Bayesian state space models. All right, so previous studies, right? We all know the experts here, Misty, Mr. Weekly, Mike, and what you all have previously looked at are pollinator limitations, reproduction, fruit and seed dispersal, and seedling establishment, though no clear explanation for the lack of successful sexual reproduction other than, right, the S allele incompatibility has been determined. Lysimachia asperulifolia has been protected for over 3 decades, yet little understanding of the species itself and how best to manage it has been gained. A population genetic survey will be the first for the species and critical for future management practices, where we're all pretty concerned about genetic bottleneck. As I mentioned just a second ago, some of you have done a lot of research on this plant, and North Carolina Botanical Gardens' very own Mike Kuntz conducted a study in 2014 where he and a team collected rhizomes of Pocosin loosestrife and propagated them in greenhouse. That research was successful with the propagation of rhizomes, and they were then translocated to other known populations, which then showed overall increase in vegetative growth, where interspecific competition was hampered either with sprays or mowing, things like that. Mike also determined, super cool, the pseudo-annual life habit in Pocosin loosestrife, right? So that's when a single rhizome produces one or more dormant below ground buds through the growing season, which then separate from the parent rhizome and continue to form new ramets the following season. So even though there's been a really good bit of research done, I think it's about time, we need to go to the molecular level now that we have the technology, right? So taking that next step towards conservation of the species, this study is using 11 unique microsatellites, which were created by a team at NC State, led by Clyde Sorensen and Elsie Youngstedt, to determine the genetic diversity among the extant populations of Lysimachia asperulifolia. Just a reminder, microsatellites are highly variable, non-coding, repetitive DNA sequences that are 1 to 6 base pairs in length. And the variations in these repetitive sequences allow information about an individual to be inferred from other members of the same population. So we collected leaf tissues across known populations in 7 North Carolina counties, including from Boiling Springs Lakes, which is Brunswick County, Croatan National Forest in Carteret County, Fort Bragg and Sandhills Game Land. That's going to include Cumberland, Harnett, Hoke, and Scotland counties, Green Swamp down in Brunswick County, Holly Shelter Game Land over in Pender County, and at Matsu, or Military Ocean Terminal Sunny Point, which is in Brunswick County. Tissue samples from Fort Bragg and Sandhills were shared by the NC State team and combined with our data set. In the field, a preliminary stem count to confirm a minimum of 20 stems with leaves in whorls of 3 or greater was conducted before collecting from each population. Stems with 2 opposite leaves or less, which I never saw, are often presumed immature and therefore will not be sampled from, or preferred to not be sampled from, depending on where you're getting your permits from and where you're sampling. So we just collected a single leaf from each stem and desiccated that in silica gel, stored in airtight containers with minimal light exposure until we went to extract DNA. When we did go to extract DNA, we initially used a QIAGEN kit, which produced lower than expected yields. We suspect there was interference from polysaccharides and polyphenols. So we switched to a CTAB protocol. Don't make me say that one all the way out. We're just going to stick with the acronym. And then we added a DNA cleanup kit post-incubation. Then we added, of course, our primers, and we amplified using PCR, right? Okay, so this is the juicy stuff. This is the juicy gossip. Our preliminary results, right? This is what we all came for today. Preliminary results are showing high rates of clonality across all 6 sampled populations in North Carolina. We knew that was going to happen, right? So our clonal richness, we can look at that first, right? That's our 3rd column in right here, clonal richness. It ranged from 0.05 to 0.24, which indicates that 76 to 95% of all samples had the same genotype. You can see here with our N, right? So from each site, we collected a different number. The G here, that's indicating the number of unique genotypes, right? So out of 20, we had 2. Out of 40, we had 4. Lots of clonality here, right? We kind of expected that. Boiling Spring Lakes and Holly Shelter Game Land had the highest rates of allelic richness, right? So that's the measure of genetic diversity, which we can use when trying to determine a population's long-term potential for adaptability and persistence. So that's right here, our AR, right? That's a good thing. We have good diversity in there, which leads into the next result, right? Our observed heterozygosity was higher than the expected, right? So there's more diversity than you would expect within these populations. Also a good thing. So the last bit of this table right here, so all the way over to the right, the FIS, right? So that's going to be the inbreeding coefficient, and all of those values are negative, which that just confirms the suspected S allele incompatibility that has been assumed since the days of Vonda Francis papers and submission for protection back in the 80s. So there it is at a molecular level. We know that it's S allele incompatible. Okay, next up, we have also observed, right, significant differentiation in genetic structure between specifically green swamp and the Fort Bragg and Sandhills game land populations. Those two populations are also very distinct from all of the other populations. And I'll explain this as I go along, right? So we have high numbers here, right? So the darkest red, that's towards the high end of our scale here. That's showing how different it is from that population. And if we go around these numbers, right? So we have 1, 2, 3, 4, 5, and then 6 down here. Those are the different populations, right? So number 1 would be Boiling Spring Lakes. Then we have number 2 is Croatan, 3 Green Swamp, 4 is Holly Shelter, 5 Matsu, and then the Fort Bragg and Sandhill samples are all going to be through number 6. So you can see the greatest diversity is between Green Swamp and Fort Bragg and Sandhills. Everything else, the other 4 clusters, the other 4 populations, essentially sit right on top of each other. So here's just a different way of viewing that, right? Number 3 in the top left, that's going to be Green Swamp. Number 6 in the yellow, all the way on the right, that's going to be Fort Bragg and Sandhills. And then everybody else is stacked right on top of each other, which is interesting to me. What's interesting to me specifically is that we're not seeing much differentiation between the Holly Shelter game land, Matsu, Croatan, and Boiling Springs populations. They're all almost right on top of each other. Specifically, Holly Shelter and Matsu, which is puzzling considering how differentiated we're seeing Green Swamp is right now. So essentially what we're seeing are 3 genetically distinct clusters among these sampled populations, which is cool. It's a step in the right direction, right? Because, you know, kind of the whole point of this and everything everybody else in North Carolina and other organizations have been trying to do is to just produce data, find things that will be instrumental in producing more comprehensive management strategies for the species. Future studies will be able to apply, hopefully be able to apply results from this research to in situ, ex situ conservation efforts in obtaining and preserving land with suitable habitat for reintroduction. Clearly, we have a lot more analysis to get through with these results, but there are some really interesting things up front that I hope you all feel the same way about. But better understanding and protecting this plant is the goal, right? And we hope to also contribute to the maintenance of the imperiled coastal plains ecosystems. So I probably talked a little too fast there. I'm just really excited and I apologize for that. Now is a great time for questions. That's it. That's everything I've got for you folks today. So thank you for sitting through that.
Well, thank you, Jess. That was fantastic. I'm not seeing any questions yet, but folks at home or in the office, wherever you are, feel free to use the Q&A tool to add any questions for Jess that you may have. Here's to kick things off. What's what's next with this project for you?
Well, we need to wrap things up, right? So finish writing that thesis. And hopefully submit for publication somewhere. And then, you know, ideally we can get the results from this shared with all of the cooperating organizations, you know, Natural Heritage Program, you guys. You actually have had a really, the Botanical Gardens has had a really large hand in conservation with this species in particular, among many others. Fish and Wildlife, we really just hope to bring this information to them for better future management practices. But we still have to hash out, you know, what everything means at this point and how best to present that to the public and to these organizations.
Right now, the questions are pouring in. Thanks, y'all. So this is from Dr. Alan Weekly. He's saying, I'm having a hard time translating this one. Reminder of the geography of the 4 populations that are genetically very similar. So I guess if you could reshare those slides showing how they are genetically similar, and then could you answer if genetics is geographically correlated?
Oof. Yeah. All right. Let me see here. I am still sharing my screen, correct? Okay.
I actually disabled or stopped your screen sharing.
There we go. Okay. So all right. So let's see. Who are we looking at, right? You got me. Geography is not what I'm good at. Geography is what Amanda has spent hours and hours, and probably at this point years, focusing on in her defense, which you were present for. So if we're thinking about Green Swamp compared to Fort Bragg and Sandhills compared to everybody else, right? Well, first of all, Fort Bragg and Sandhills, that's in a separate part of this floristic region, right? So you're up in the actual sandhills, you have a higher elevation. As far as the geography, again, this is something that I need to write into my thesis, and I'm not great at it just yet. But I'm kind of confused with the findings of Green Swamp being so distinct from everything else, with the exception of the sandhills populations, given the location. Like evolutionarily speaking, you would figure perhaps Holly Shelter and Green Swamp would be more closely related. However, Holly Shelter is more closely related to the populations down in Matsu and Boiling Springs, and Croatan, which is even stranger to me. So I don't have a really great answer to that. The geography is something I still need to look into and lay out in the thesis. But, just geographically speaking, as far as localities go, this is strange to me. This is an interesting, I don't know that the geography is related at this point. We're not really seeing that. However, mind you, that's within the parameters of this study. So, we have a total N of under 400. You can always increase your sampling size, your sampling area. We didn't go down to South Carolina, things like that. But with the results that we do have, I don't think that geography is correlated. But I do obviously need to learn that geography and write it down, don't I? Did that help? Did that answer to the best of my abilities? Anywho, did that?
That was a great response, and it actually leads into another great question. You spoke about the parameters of your study, but what other questions or sampling strategies would you like to see done with this species? With folks using your research or just taking their own research, taking their own spin on this?
Yeah, replication in science, right? It would be great to see a study like this, right, replicated at a much larger scale, right? So obviously the South Carolina populations need to be included for a more encompassing measure of the diversity that we're seeing, right? Crossing state lines, that's quite a big distance. And if we don't think geography is related, well, going to the next state over would help answer that question a bit, maybe. So expanding upon this to triple maybe the sampling size at a minimum. Funding is of concern always and forever when you're doing this kind of research. So if some organization could get a grant to just do this at a larger scale, and perhaps for a longer time, that would be really interesting to me.
Makes sense. Yes. Funding is critical to this work.
Yes, it is.
And Alan shared a couple of positive comments in response to your response to his question. Perhaps the 4 populations versus Weenstop may be a little bit more inland, or there's just some weird founder effect or a bottleneck of past events.
Founder effect is something that I'm really thinking about right now. That's an excellent, thank you for sharing that.
And then we did have a question in the chat. Someone asked if the botanical garden is seed banking from these populations. I had to ask our director of conservation, Johnny Randall, for the answer. He's sharing that we don't, because so few of these populations do produce seed. So hopefully,
and just
in
as an
future...
aside, to his final comment that so few populations are producing seed, the only population that I sampled from and I saw fruits and evidence of seeds were at Holly Shelter. That's the only, out of all 6, that's the only place that I saw any evidence. And it was in one of the smaller populations within Holly Shelter, strictly the power line.
And as I saw in your bio, you are a future conservation biologist. What recommendations do you have for future conservation efforts of the Pocosin Loosestrife?
Yeah, so how are we directing our energies here? What are we looking at? One of my biggest concerns, and this is in conservation biology in general, this is a standard, is to do stem counts, to look at viability of a population, health of a population. Unfortunately, I think with clonal plants, and in particular with this plant, we just need to throw that out the window. We need to find a different baseline. We can't really, I don't think we can use that blanketed approach for clonal organisms, but in this particular situation with this species, I don't think it's an effective measure of population vigor. I just don't think that that's fair, because we know that from year to year it changes. We know that it varies with and without fire. We don't really see correlation with prescribed fires and increase in vegetative or floral propagation. We need to reevaluate these kind of basic assessments we've been using in looking at Lysimachia populations. So just furthering that molecular perspective, I think it's an absolute must. We have to kind of start going in a different direction with these known populations while we still can.
Great, thank you for your thoughts on that. This is all above my head, so I'm just gonna smile and nod. It's a great approach. Listen to her. We have one more question about hybridization of Lysimachia species. Does it hybridize with other?
That's Suzanne. Please say it was Suzanne. It was, wasn't it?
It was not Suzanne.
Oh. So this is actually something on the front end of this degree. So I started in fall of 2019. And then the pandemic hit right when I was about to start applying for everything and sampling. But I bugged the ever-loving life out of Misty Buchanan at the beginning of this. And she presented a theory of possible hybridization between Lysimachia asperulifolia and Lysimachia lumicii. And that would be another molecular avenue to go down. I think hybridization is very interesting thought here, and something that's come up consistently through all studies of this species from Vanda France on, has been inconsistencies with floral morosity, fruit production, seeds. There's just nothing that's the same in any of these distinct populations morphologically. And seeing such variety would really make one question if hybridization is a possibility. So again, just going down that molecular avenue, I don't have an answer to that with the type of research that I've done or that we're trying to wrap up right now. But I think that would be a wonderful addition to, again, the conservation, the management protocol, seeing if any of these populations are in fact hybridizing with close relatives.
For that. Not seeing any more questions at the moment, so if you have any, feel free to add those. In the meantime, when are you expected to finish your degree, and how can folks get in touch with you with job opportunities? I know there's some great organizations here in the webinar that
There
maybe
are.
as
This is
impressed as
probably
I
why I
am.
was talking so fast the whole time and nervous about everything. So I'm hoping to apply to defend this summer, so sometime before August. And after that, yeah, communication. I can, I'm gonna pop my personal email in the chat, I guess. Is that how this works? Is that what you do?
You're welcome to.
I was just hoping to bother, like I did with Misty at the beginning of this degree. I was hoping to email and call people until they had to talk to me. But I'm sure there's a more appropriate way to do that. So there's my email, just in case anybody is interested in reaching out. I do really want a job, that's for sure. And I think more research on this plant in particular is definitely needed. But I think we're taking a good step in the right direction here. So continuing this would be a really interesting opportunity if anybody were to have the funding for it.
Well, seeing no more questions coming in, I will invite Joanna back on to close us out.
Thank you.
Thanks again, Jess.
Jess, thank you so much for being here. We really appreciate it so much. That was a great presentation.
Thank you for having me.
Yeah, and I wish you all the best moving forward.
Thank you so much.
Job on the horizon. So
thank you.
Well, thank you to everyone else for joining us here today. And I'll ask too, if you're not currently a member of the garden or the Friends of Plant Conservation, we hope you'll consider becoming a member. Membership is critical to both of our missions. Thanks so much.